Welcome to the Classics in Immunology Journal Club. And I just want to say up front, if you like this journal club and you like this particular video, then go on the website, the YouTube website, and like it and click on the little bell so you get notified when I put another one up. And then scroll on down to the bottom and go to my website. There's all kinds of information there about cytokines and T cells and immunology. So today, last week I told you that 1970 was a banner year, and we're still doing another article from 1970, and also an additional article from 1971. And these two articles are the beginnings of interleukin-1 and interleukin-2 that really were not defined until another decade later. But if you use a little bit of imagination, and you can really understand where the nidus of all this business about cytokines and lymphokines, or if you prefer LYMPHODRECK, came from. So the first paper is by a fellow by the name of Fritz Bach, and he had students and postdocs working with him, Barbara Alter, Susan Solliday, David Zoschke, and Martin Janis, and it comes out of the University of Wisconsin. And it was published in a new journal called Cellular Immunology. So, as I've said previously in some of these videos, that the real immunologists were immunochemists, and they were trying to find out what the nature of the immunoglobulin antibody molecules were. And so they were hardcore chemists who fashioned themselves as the real immunologists. And so what the new immunologists, or I guess the false immunologists, if you want to say that, they started their own journal in 1970, and that was Cellular Immunology, and this is in the first volume. And the title of the article is Lymphocyte Reactivity in Vitro, and this is number 2 of a series of articles. And it's entitled Soluble Reconstituting Factor Permitting Responses of Purified Lymphocytes. And a little background into Fritz Bach. He's an interesting guy. He was born in 1934 in Vienna into a Jewish family. And so in 1938, when the Kristallnacht happened and the Nazis started destroying everything that was even related to Jews, his parents got out, and they went first to the UK and then ultimately emigrated to the United States in 1949. So Fritz was only 4 when they went to the UK, and they stayed there for, say, like a decade or so, and he was 15 then in 1949 when they emigrated to the United States and ended up in Vermont, up near Burlington, where Fritz grew up the rest of the way. Ultimately, he went to Harvard College, graduated in 1955 in science, and then went on to Harvard Medical School and graduated in 1916. Then he went to New York and to NYU, where he came under the influence of one of the old guard scientists of that generation, Lewis Thomas, who was the head of the pathology department, who was very influential for a lot of individuals that ultimately ended up in immunology. And it says in one of the obituaries of Fritz, and he's just died recently in the 2000s, he was tremendously influenced when he was at NYU by a lecture by Sir Peter Medawar, who came to New York and gave a lecture about this transplantation situation of skin graft rejection and that sort of thing. And so he decided to devote his career to that, which he did. And so in the early 60s, as you recall, Peter Knoll discovered that lymphocytes were not just sort of boring end-stage terminally differentiated cells, but they could actually proliferate. They could replicate their DNA and undergo mitosis as long as you stimulated them with something. And that's something that Peter Knoll figured out was phytohemagglutinin, or PHA. And then soon thereafter, within the next year or 2 or 3 or 4, multiple investigators in the early 60s showed that specific antigen would do it. And in addition, specific histocompatibility antigens. So if you mixed lymphocytes together from 2 different individuals, and you inactivated one of the individuals, they became the stimulator cells, and the other ones were the responder cells. You get a brisk proliferative response out of that, and that was ultimately called the mixed lymphocyte culture. And that was first described from McGill in 1962, 3, 4, somewhere around in there, by Barbara Bain, who was in the hematology department. Well, Fritz got onto this, and he thought, well, this is the greatest thing since crushed ice, because what you can do is you can do these mixed lymphocyte cultures in vitro before you try to graft a kidney onto somebody to see, what the reactivity would be. And so he wanted to use that test, this in vitro lymphocyte proliferation assay in a mixed lymphocyte culture, in order to test people as to their histoincompatibility or compatibility, to try to diagnose before the transplant whether the transplant would actually take. And so he was beginning things then in the late 60s. What had been at that point in time, in the introduction of this paper, what the authors talk about is number one, that it had already been shown only a couple of years previously, in 1968, by Joost Oppenheim and others, that if you'd use purified lymphocytes and did a mixed lymphocyte culture or a specific antigen, none of the cells would proliferate. And then if you added back into them what they thought were the macrophages, then they would proliferate. So that was already known. And so what this paper is about is trying to understand what contribution the macrophages give to this whole process. And the experimental system was simply taking lymphocytes from one individual or another individual, purifying, getting rid of the so-called glass adherent cells, which were the macrophages, and then culturing purified lymphocytes versus unpurified lymphocytes, and assaying them for the incorporation of radiolabeled thymidine, tritiated thymidine, in the newly synthesized DNA, which is then a precursor. Once you replicate your DNA in a cell, then that cell can undergo mitosis and actually divide, and that's actually then proliferation. Now, they didn't really know exactly what the macrophages might be doing, but they wanted to find out whether or not the macrophages made anything and maybe released some stuff, antigens, enzymes, whatever, nutrition or hormones that would help the lymphocytes recognize the antigen and proliferate. And one of the things that's interesting about this paper is that in the introduction, they point out, they said, we've been talking about macrophages and adherent cells, but there's a gamish of cells in the peripheral blood floating around, a lot of white blood cells, different kinds of white blood cells. There's, as I've said before, there's macrophages, and then there's microphages, the polymorphonuclear leukocytes, and so forth. And we don't really know yet. We're calling them macrophages, but we don't know what's really going on, what kind of cells there are. And they also say in the introduction, they said, you have to remember now, back 5 years previously, in 1965, and we've already reviewed those papers here in the Journal Club, there was such a thing described as blastogenic factor. 2 papers in Nature in 1965 from McGill University in Canada, Montreal, described if you cultured lymphocytes, you did one of these mixed lymphocyte cultures and let it go, and the cells would proliferate. And then if you took the supernatants from those cultures and got rid of the cells by centrifuging them out, then you could take that so-called conditioned medium and put it on fresh lymphocytes and fresh mixed lymphocyte cultures, and you could get a marked heightened response, potentiated response. And that was known. What wasn't known was what cells are making this, whatever this stuff is, this factor, blastogenic factor. And it was for sure wasn't known what the molecular composition of this was, an activity. The other thing that people had started to wonder about was whether or not there was only one factor, or maybe there was other factors there. depending on how they did their assays, some people started talking about a potentiation factor. That is, not only did you get some activity that they called blastogenic factor, if they got more activity, more proliferation, they said, well, maybe that's due to another factor. It's a potentiation factor. And they also found in different experiments and so forth, evidence that there were inhibitory factors in this conditioned medium. And that if you had the right proportion of stimulatory versus inhibitory factors, you might not get any effect whatsoever. So it was, talk about phenomenology. This is really phenomenology par excellence, essentially. The Fritz Bach's lab started to look at what these macrophages might be doing. And so they were culturing the adherent cells, or the so-called macrophage things, to see whether or not they would release anything into the conditioned medium. And they found evidence for what they called then a conditioned medium reconstituting factor, CMRF. That's a mouthful, and it never really caught on. So, you gotta, it's just like bestsellers. If you look at the New York Times bestseller list, you can't have two words in a title of a book. You gotta have just one that rings. And then people can remember that. Most of the time, they don't even remember that. So same thing with factors. So that was in the intro, and that was in the background. And so in the materials and methods, what they did was they would draw white blood cells from blood, from their lab mates, essentially, put them in tissue culture 199, and they didn't use fetal calf serum, which is really important. They used normal human serum or plasma that they got from the patients, from the people that they drew, that they used to get the white blood cells. They used the same plasma, so that they didn't have sort of a confounding factor of the fetal calf serum might be doing something in their cultures that they didn't understand or didn't know about. And so they cultured the cells from one individual, the responding cells, and around 2 × 10⁵ cells, 200,000 cells per milliliter. And then they put in stimulating cells that they had inactivated with a chemical called mitomycin C. You could also use radiation to block them from proliferating. So those became the stimulating cells. And they were at twice the concentration. They were at 4 × 10, 400,000 cells, 4 × 10⁵. They would culture those cells then for a week, for 7 days. And then overnight, they would throw in tritiated thymidine, which would be incorporated into any cells that were replicating their DNA at the time. And that was the way you did these experiments. In order to make their factor, their adherent cell factor, their CMRF, they took the adherent cells from one of these leukocyte preparations, glass adhered the cells, washed off the lymphocytes, and several times, and then cultured the cells for 3 days in tissue culture media plus the plasma, the autologous plasma. So that was it. That was the sequence of events, and then they would take this conditioned medium and add it to their mixed lymphocyte cultures to see whether it did anything, particularly with comparing the purified cells, lymphocytes, versus the unpurified lymphocytes that already had the macrophages there. And so this paper in cellular immunology is, it's, even though it's in the Classics of Immunology Journal Club, they did this paper in a hurry. And it's one of the reasons is they had a competitor that they were dealing with, and the competitor was Richard Dutton, who I talked to you about the Michel Dutton assay. Well, that's the second paper we're going to review today. And they were doing very similar experiments. And so I think Fritz was in a big hurry, because in this paper, in the results section of the paper, there's only 3 tables. And the first 2 tables are just individual experiments. The last table is a compilation of 10 different experiments. So at least they did it more than once. But it's not one of those rigorously defined and where they've beaten the dead horse to death. This is sort of just the beginning of this sort of stuff. And in the first table, what they found is that when they added their factor, their CMRF, to purified lymphocytes in the mixed lymphocyte culture, that without the factor, they got 100 counts per minute. So the nice thing about the 3,8-5-adenoids is that you got a quantitative readout that was not subjective. It was very objective. You stick the stuff into the radioactivity counter, you get counts per minute out. So with just the mixed lymphocyte culture alone, that did not proliferate without any macrophages, they only got 100 counts per minute incorporated. Whereas when they added their factor, they got 19,000 counts per minute incorporated. So that was pretty dramatic, you know. Then they did an experiment in table 2 where they compared unpurified mixed lymphocyte cultures to purified mixed lymphocyte cultures. And they added their conditioned medium, and they wondered whether or not there was any blastogenic factor activity. Well, the way they defined that was an operational definition is that if it worked to enhance the proliferative response of unpurified cells in an MLC, that was a blastogenic factor. That's what they decided to call blastogenic factor. And then they also had in table 2, you know, the other control of the purified lymphocytes for their reconstituting factor. And it gets a little bit, you know, the operational definitions of these factors is sort of suspect. Then in table 3, they did other experiments where they diluted out their conditioned medium factor, and they found that they diluted out 1 to 10, 1 to 100, 1 to 1,000, and the activity fell off after 1 to 100. Well, that was pretty something, because that meant that there was a concentration of the factor in the original conditioned medium was pretty high. So in the discussion of their paper, they point out several things that are really, I think, striking. One is they said, you know, there could be 3 activities in our conditioned medium, or there could only be 1. And it could be depending on how you assay them, essentially. And they went on to say, I had to read this, so to quote, that any of the activities it's not proper to conclude that any of the activities represent different molecules. They may simply appear different because we've changed our operational definition of how we assay for them, how we measure them. So these kinds of things, that's basically about all there is in this paper. And these are the kinds of things that came under scrutiny by the immunochemists, for example, and the other real immunologists. And that's one of the reasons that people started saying, whatever, you know, it's LYMPHODRECK. It's not good science, essentially. So the next paper was published the very next year by Dick Dutton with a postdoc, Michael Hoffman. Their paper was in Science, so they didn't have to resort to the new upstart cellular immunology journal. They got theirs in one of the best, Nature and Science. And that was published in 1971. And Dick Dutton was at the University of California at San Diego. And we've gone over his background and so forth. He was a Brit that became a colonialist and came over to our country. So in the introduction, they say basically that Don Mosher, whose paper we reviewed, showed that if you took macrophages out of the Michel-Dutton system, where we're trying to do an in vitro stimulation of non-immunized spleen cells from mice with sheep red blood cells, that the purified splenocytes, lymphocytes, without the macrophages in there, they didn't get a response. And that's what Don Mosher reported. Well, so they said, well, maybe let's find out what these macrophages are doing. And there were several different possibilities. But they wondered how, for the first time, they started talking about a helper effect that the macrophages were doing. And they wondered whether or not if they tried culture supernatants, just like Fritz Bach's CMRF, whether or not that could replace the macrophages in the Michel-Dutton system. So you see what's going on in cellular immunology in this beginning part of it, or beginning era, was there basically just 2 or 3 assays. You had radiated thymidine incorporation after a mixed lymphocyte culture or a specific antigen like a TB antigen, or you had the readout of antibody forming cells, plaque forming cells in the Michel-Dutton system. And as time went on, and we'll get into it, you could generate from a mixed lymphocyte culture, you could generate cytotoxic cells that you could assay in the cytotoxicity assay that we talked about, the so-called chromium 51 release assay. So that was it. These basically are the bread and butter of cellular immunology in these early days. So in the materials and methods of this paper, they used Eagle's minimum essential medium with 5% fetal calf serum, and you had to select the good batches of fetal calf serum. Nobody seemed to know what that was, but if you screened a whole bunch of different fetal calf serums, some gave you a better response in your assay of the number of plaque forming cells than others. And just as an aside, when I first got into the business around 1970, I looked at both the thymidine uptake assay and I looked at the cytotoxicity assay, and I looked at this plaque forming cell assay, and I decided to myself, I distinctly remember it, I thought to myself, they don't know what's going on in this plaque forming cell assay, and this whole business with the fetal calf serum and the good batches and the bad batches, that bothered me. And so I decided not to deal with that. I put my money on the thymidine incorporation assay. So they would take their spleen cells from mice, and they would get macrophages from the peritoneal cavity. They would squirt into the abdomen tissue culture medium, slosh it all around, and then aspirate it out. And they could put that down onto plastic or glass, and that's how they could get their macrophages. They purified the lymphocytes and got the macrophages out by passing the lymphocytes, incubating all spleen cells with glass beads, which did the same thing. The adherent cells adhered to the glass beads, and so you could take them and get the lymphocytes out from that and leave the other stuff behind. They used 2 different antigens. They used sheep red blood cells and also burrow red blood cells, and they didn't really cross-react at all, essentially, is what they said. And so in the results of this paper, so they tested the purified spleen cells with and without macrophages and with and without adherent cell supernatants to see whether or not they could replace the macrophages or the macrophage activity with the soups. In the very first table, they show that when they removed the plaque forming, the attached cells, and they used purified lymphocytes, that the number of plaque forming cells in the unpurified preparation was 2,500, but in the purified population, it was only 168. And that was the mean. Those are just the mean numbers of 4 or 5 different experiments that they put into the table 1. We're still doing tables in biology at this stage of the game in terms of representing the data. People didn't start yet using figures, which are much easier to gain the major, the bottom line from, and the conclusion as to whether the experiment worked or didn't work when you're reading through the text and you're trying to decide whether or not figure 1 really shows the data that what they're saying in the text or not. Tables are much more tedious to discern. So then also in table 1, they used 24-hour supernatants from macrophages, and it restored the plaque forming cell response. And this is what they were looking for. And even when they diluted it out to 100 or 1 to 270, they still got a response. And they said that when they tried to destroy the activity by heating the supernatant to 57 degrees for 30 minutes, or by passing it through a millipore filter, a 0.2 micron filter, that it didn't remove the activity. The activity was in a small enough molecule that could pass through the filter. The thing, they did some experiments with sheep red blood cells that they packed into, or they put into a test tube and then incubated their supernatant with, and then for a while, and then spun out the sheep red blood cells and took the supernatant, and they said it could be partially removed. They thought it could be partially removed based on their experiments by the antigen, by sheep red blood cells. And they also said if they harvested those sheep red blood cells that had been exposed to the conditioned medium, and they could use them in subsequent experiments, and they would be antigenic for the plaque-forming cell response. And the data in this paper are not very solid or convincing that there was anything going on from these manipulations. But that was it. So that was the paper. So there was these two different schools, the thymidine incorporation people and then the plaque-forming cell people. And they were both saying, well, these macrophages are releasing something that seems to be, it might be an interesting thing to look at. They speculate, Dutton and Michael Hoffman do, as to what this activity might be due to. And so first they say, well, maybe it's an antibody. Maybe the spleen cells are making an antibody to the sheep red blood cells. I don't know why that would function in that way, but that was one of the possibilities. And then the other thing they thought, well, maybe the macrophages are releasing enzymes that do something to the sheep red blood cell membranes, and that makes them better stimulators of the purified lymphocytes. And then finally they say, well, maybe it's just nutritional, like carbohydrates and fats and proteins, and makes the cells happier and grow better and so forth. Or finally, maybe they said maybe it's hormonal. What a hormone is specific molecules that are usually produced by glands in our body. We have different kinds of hormone-producing glands. They make these molecules, they secrete them into the bloodstream, and then they travel to other places in the body and stimulate other tissues and organs by binding to specific receptors. That's what a hormone is. And that was probably what was going on. We'll find out as time goes on. So I think that's right. They go on to say then in the interpretation, maybe we've rediscovered the wheel. We really rediscovered blastogenic factor, whatever that was. But we don't have any idea what the molecular nature of this thing is. And I think in retrospect, looking back now, so we're looking back 50 years ago when they were doing these experiments, the big bugaboo in all of these experiments in the Michel Dutton system were the special batches of fetal calf serum. That's number one. Number two, the number of plaque-forming cells that they were finding, and they reported them out as the number of cells in the culture per million spleen cells. And so when they get 1,000, that's only 0.1% of the whole total. And secondly, when they looked at not what they thought was a primary immune response, but rather a secondary immune response, taking spleen cells from animals that had been immunized in vivo with sheep red blood cells, they had several 100,000 plaque-forming cells. So these guys were working way down at the minimal level of things going on in their cultures, and because of the fact that they were using fetal calf serum and special batches of fetal calf serum, I think that most, if not all, of what they were seeing was simply the lipopolysaccharide, the endotoxin from gram-negative bacteria that were contaminating the fetal calf serum. And that's because LPS is a non-specific mitogen stimulating the B cells in the mouse. It doesn't do anything to human cells, but in the mouse, it causes the B cells to proliferate. But anyway, that was the beginning of a whole area of cellular immunology and science, and we'll find out more about how this goes on as time goes on, and if we're still alive as we go through all these experiments. So don't forget, like this video, that'd be helping. That helps the YouTube people really like that. And then also click on the bell so that you get notified when I put another thing out, and scroll down and check out my website. You can see all the papers from the classics from the last 200 years there. So thanks a lot. It's been good.